So if we talk about proteobacteria, so most of them, they are gram negative and chemoheterotrophic bacteria. And what is this term chemoheterotrophic bacteria? We have discussed it in the nutritional types, and it is believed that they are chemoheterotrophic bacteria. Some of them, all of them are gram negative, but some of them are photosynthetic as well. So that's why it is believed that they have some common photosynthetic ancestor. And it is the largest taxonomic group of bacteria. हम डिस्कस कर रहे हैं सेकंड एडिशन का वॉल्यूम 2, राइट. वॉल्यूम 1 भी हम पांचों वॉल्यूम डिस्कस करेंगे, बट नाउ वी आर डिस्कसिंग वॉल्यूम 2, राइट. ग्राम नेगेटिव proteobacteria, which is a volume of gram negative organisms. So all in all, 3 volumes they belong to gram negative organisms, 2 volumes they belong to gram positive organisms. So ये volumes हमें कहाँ help करते हैं? For example, if you have identified any species, bacterial species, so to start with, what you will first do? You will first do definitely the gram staining, right? Once you know the gram reaction, then you will find out the further details in those 2 volumes which are gram positive, right? So you will search for that organism not in volume 1 and 2 and 5, but also in volume 3 and 4. Later on, while discussing or while discovering different properties of that unidentified strains, you can match that out of those 2 volumes to which it is matching the most. And whenever you will perform a 16S ribosomal analysis, then you will definitely get to know about its complete identification. Okay, so few are, I told you that are photosynthetic, and this relationship there, it's based on ribosomal RNA studies. And the name proteobacteria, why it is given the name proteobacteria? Because it is based on a Greek god which is known as Proteus, and the Proteus means that it can assume many shapes. So in this group, because you can find different varying shape organisms that are of different shapes. So you may find curves, you may find rods, right? You may find some spiralums, so they can assume many shapes. That's why the group is named as Proteobacteria, right? And again, now this is one of the largest group. So in this group, in this volume, this volume is divided into 5 different classes, right, which is named on some letters: ALPHA-PROTEOBACTERIA, beta proteobacteria, gamma proteobacteria, delta proteobacteria, and Epsilonproteobacteria. So I'll discuss one by one. So alpha proteobacteria, they are unique in that sense that they only require very low levels of nutrients to grow. And another unique property is that they have some protrusion, means they are not that much symmetrical in the sense that they do not have smooth surfaces. Rather, there are some protrusions like some birds on the surfaces, and at some times there are some stalks which are known as prostheca. I'll show it in the picture later on in the next slide. And a very important characteristic is that they are agriculturally important because they are capable of inducing nitrogen fixation. So they play a very much important role in the cycling of the nutrients. So soil की जो nutrient cycle है उसमें बहुत important role play करते हैं. They are mandatory to be present in the soil because they provide plants nitrogen, because they are capable of inducing nitrogen fixation. As you all know that plants cannot fix nitrogen, so they are always in need of those organisms present in the soil, microorganisms that are capable of fixing the nitrogen. So yes, these organisms can fix nitrogen, and there are several plant and human pathogens. Now I'll start from Pelagibacter. It is one of the group that belongs to aquatic system. So it is most abundant microorganisms on earth, and definitely most of the earth is aquatic. So it is basically ocean based organism. So it is one of the most abundant microorganisms in ocean. So that's why we can say that it can be the most abundant microorganism on earth. So it is one of the unique feature of this particular group. So this was first discovered by a technique known as fish. Oh my God, yeah, look. Anyways, so it was first discovered by the technique fluorescent in situ hybridization, which is also known as fish. And the group that was first, the candidate that was first discovered, it was named as Pelagibacter SAR11 because of the reason that it was first discovered in Sargasso Sea, and 11 it designates the species number. And I told you that there are different bases for naming. So one of the example is this Pelagibacter SAR11. One of this member of this particular group, Pelagibacter, एक species है इसकी, that is known as Pelagibacter ubique. This is the candidate that can be successfully cultivated in the lab, right? If one of the member is discovered, it doesn't mean that it can be cultivated, right? That's why it was discovered by the use of this fluorescent in situ hybridization technique. ये क्या होती है इसके बारे में आप मुझे WhatsApp ग्रुप पे बताएंगे, because now you will be able to understand the things. इसका जर्नल जो मैकेनिज्म है वो हम WhatsApp ग्रुप पे डिस्कस करेंगे. आपका असाइनमेंट है कि what is the basis of this technique and how they discovered this particular organism. So this group, most of the members, they are not culturable because because they have very unique combination. They require very unique combination of nutrients which we are unable to provide them in the lab. So that's why we cannot cultivate them. So but this is the member that can be successfully cultivated. It's the first member, and it regulates the earth carbon cycle. How it regulates the earth carbon cycle? Because I told you that it is one of the most it belongs to one of the most abundant group of microorganisms in the earth. So why it is called, that's why it is called as Pelagibacter ubique, means it's ubiquitous everywhere in the ocean. And it's how it is actually surviving in so much abundance in the ocean, because its genome has been sequenced, and it has only few genes. Few genes means the smallest number of genes, and it is a free living organism. It is not parasitic in nature. It is surviving. So it is maintaining it is maintaining all its nutritional requirement itself. So for a free living organism which has to prepare everything, which has to manufacture everything on its own, the number of genes which are required is much higher than those which have some parasitic nature or symbiotic nature. right? For a free living organism, and everything is self-dependent, so number of genes which is required is high, but the uniqueness of this organism is this, that with this small number of genes, it is surviving as a free living organisms, right? So the reason that how it is surviving, because its size is extremely small, right? So it's small size and its minimum genome, it is actually giving it a competitive advantage for survival in the low nutrient environments, right? So this is the reason that how they are behaving as the most abundant microorganisms, because of the size and because of the smallest number of genes, right? Because gene expression also requires energy, so more gene, more energy consumption. So less number of genes, less number of less quantity of energy consumption. So I hope that you can now relate that how they are maintaining their abundance in the uh, ocean environment. So the other thing is other candidate, yeah, other species is the Azospirillum. As its name indicates, the word azo, it refers to the ability to fix nitrogen. कहीं भी आपको word azo नजर आएगा तो it's somehow it indicating this character of that organism, that it will be capable of fixing nitrogen. Spirillum, it indicates its shape. So क्या है so azospirillum, I told you, it definitely, once they are fixing nitrogen, they must be in close association with the roots of the plants. Definitely they are in soil because they are fixing nitrogen. So they are somehow, they are dependent on plants because they use nutrients excreted by the plants, means they somehow maintain some symbiotic relationship with plants, and they fix nitrogen for them. So commercially, आपकी अगर इसको commercial importance आपको समझनी है तो आपके पास ये यहाँ पर pictures में visible है कि there are different companies that can that are commercially using this organism as a biofertilizer, because biofertilizer helps in improving the soil fertility by making the nitrogen content available. So they are serving as a biofertilizer. So they are different this culture is being packaged in commercial packing and it is used in the farms, and as names indicate, as a spirillum के name से जो है वो ये जो organism है इसके जो members हैं, they are being marketed. So commercial importance भी इसकी है because this character is nitrogen fixing character is making it a good candidate for serving as a biofertilizer. So as its name indicates, a nitrogen fixing biofertilizer, azospirillum, and it is for which group? I told you that especially for tropical grasses and sugarcane, and sugarcane is one of the big industry, right, leading to further many industries. I am recording this lecture, no worries. बाकी क्वेश्चंस मैं भी देखती हूँ क्या कह रहे हैं आप लोग. Then we have another group. हम इस वक्त जो बात कर रहे हैं, मैं बार बार बताती चलूँ, हम अल्फा प्रोटियोबैक्टीरिया के कुछ प्रोमिनेंट मेंबर्स की बात कर रहे हैं ताकि आपको पता हो कि हर ग्रुप की जो इम्पोर्टेंस है उसमें कौन कौन से ऑर्गेनिजम्स जो है बिलोंग करते हैं, उनकी क्या कमर्शियल और क्लिनिकल, या तो उनकी कमर्शियल इंपॉर्टेंस है जैसे मैंने पीछे डिस्कस किया, या उनकी क्लिनिकल इंपॉर्टेंस है जो अभी हम आगे डिस्कस करेंगे, या उनकी इकोलॉजिकल इंपॉर्टेंस है जो अभी हमने डिस्कस किया कि फ्लैजीबैक्टर हो गया या एजोस्पाइरलम हो गया. इसको इकोलॉजिकल इंपॉर्टेंस भी है, कमर्शियल इंपॉर्टेंस भी है. इकोलॉजिकल किस तरह से है कि नाइट्रोजन फिक्स कर रहे हैं, मेंटेनिंग द नाइट्रोजन साइकिल, राइट. और कमर्शियल किस तरह से है कि इनको कमर्शियली भी यूज किया जा रहा है. दे मे बी additionally added in the soil for improving the fertility. Now another member is of commercial importance is Acetobacter and Gluconobacter. So as its name indicates that it is Acetobacter, it is able to convert. Both the species have this character that they are aerobic organisms, gram negative, that can convert ethanol into acetic acid. So definitely once they are converting ethanol into acetic acid, so their commercial importance is evident here, that you can witness that these organisms are used. So आपका भी एक असाइनमेंट है, आपने थोड़ा सा इसको सर्च करना है कि कौन कौन से विनेगर्स इस वक्त अवेलेबल हैं जिसमें ये स्पीशीज जो है वो एज अ कल्चर यूज हो रही है फॉर कन्वर्जन ऑफ इथेनॉल इनटू एसिटिक एसिड. आजकल केमिकल कन्वर्जन भी हो रही है और बायो कन्वर्जन भी हो रही है यूजिंग दीज ऑर्गेनिजम्स. तो हम टारगेट कर रहे हैं दोज कन्वर्जन्स ऑफ इथेनॉल इनटू एसिटिक एसिड that are actually utilizing these two organisms or the species of these organisms. Right, now come to the member that has some clinical importance, which is known as Rickettsial. Rickettsial का तस्करा हम पहले भी कर चुके हैं और वहाँ हमने जो तस्करा किया था वो Rickettsia और Chlamydia का साथ किया था when we were comparing bacteria with viruses and this Rickettsia and Chlamydia. So Rickettsia, Chlamydia और क्या कहते हैं उसको, Coxiella, yes, Coxiella, Berneti का नाम आपने सुना होगा. These three groups, they were grouped together in the first edition of Bergey'S manual because they share this common property that within the bacterial group they are obligate parasites. They are requiring a living host to replicate, to multiply. So without that they are not able to survive, right? But Rickettsia का एक और अब जो है इनकी classification, because now it is based on 16S. So Rickettsia is in this group, in ALPHA-PROTEOBACTERIA. Chlamydia जो है वो हम next उसमें पढ़ेंगे, एक next शायद Epsilon Proteobacteria में, और Coxiella का तस्करा in between कहीं आएगा. So now they are separately grouped. So basically Rickettsia का आपने एक और जगह तस्करा पढ़ा था और जब मैंने आपको वो पढ़ाया था Rickettsia प्रोवेजिकी के नाम से हमने डिस्कशन किया था कि उसका 16SR DNA जो मैच कर गया था with that of mitochondria, अगर आपको थोड़ा सा याद हो तो वहां पे भी एक टेक्सोनोमिक सिग्निफिकेंस भी इस ग्रुप की मेंबर्स की है. ठीक है, दे आर रॉड शेप, दे आर कोकोबैसिलाई, ग्राम नेगेटिव. ये तो एक जर्नल प्रॉपर्टी है जो हमने पहले से डिस्कस की है. ठीक है, तो एक डिस्टिंग्विशिंग फीचर इन रिकेट्सियास का ये है कि दे हैव टू होस्ट मोस्टली. They are transmitted to humans by bites of insects and ticks. So their life cycle, it revolves around two hosts, right? One is mostly insects and one is the human being. So human is usually the second host, and one of the most prominent feature of this Rickettsia is that they enter the cell. It's not that much prominent. जो मैं बताने जाऊंगी उसकी बात मैं करी थी कि they enter any cell by means of phagocytosis and they can be cultivated artificially in the cell culture and or stick embryos. Means if you want to cultivate any rickettsial species, you want to work with any rickettsial culture in your lab, so you cannot cultivate them on agar plates. Definitely, I told you, because they are obligate parasites. So you need to cultivate them artificially. You can cultivate them in lab, but in the cell culture, in cell lines or in chick embryos, because I told you that they need host. So and a lot of diseases they cause in the human beings, but all of these diseases they are grouply called as spotted fever group, क्योंकि इस रिकेट्सिया की जो खास एक स्पेशल फीचर है इसकी पैथोजेनेसिस का कि it makes your capillaries very thin, means it increases the permeability of your capillaries, blood capillaries. So the disease it appears in the form of rashes, right? So that's why it is called as a spotted fever group. Spotted rashes are there because at different places of a skin spots are visible. That is because of the reason I told you that there's this infection, it increases the permeability of the blood capillaries, right? So along with the fever, so this is one of the chicken embryo cell and masses of the rickettsial nucleus and scattered rickettsia. This is how they are spread in a cell. They how replicate in a cell. So there are different rickettsial infections. Epidemic typhus, it is caused by that candidate we discussed previously in the previous classes, by Rickettsia provesici, and it is transmitted by lice. मोस्टली ये जो रिकेट्सियल इन्फेक्शन है आप इनका मुझे आपको ये असाइनमेंट भी देना था कि आप इनका रीजन जरा ढूंढेंगे कि वेयर दे आर मोस्ट कॉमन, राइट? So another infection, a common infection or the most famous infection is endemic murine typhus. Now the word murine, it refers to rats. So caused by Rickettsia typhi, and it caused by rat fleas. And another fever is Rocky Mountain spotted fever, and it is transmitted by ticks. So insects and mostly ticks, they are responsible for Rickettsial infections. So I told you that they increase and they damage the permeability of the blood capillaries, and it results in the characteristic spotted rash. So whether it is epidemic typhus, it is murine typhus, or it is Rocky Mountain spotted fever, so all of them are characterized by a type of rash. So that indicates a rickettsial infection. Another member of clinical importance is Ehrlichia, although its infection is not that much common, but definitely it happens. I told you a picture of Ehrlichiosis. So again, it's Rickettsia-like bacteria, gram-negative, and they live obligately within white blood cells. This is one of the unique feature of this particular group, that white blood cells usually defends your immune system. So if any organism is capable of living obligately within your white blood cells, so you can imagine the capability of that organism. So it is also transmitted by ticks to humans, and it causes ehrlichiosis, and it is some time of fatal disease. Okay. Okay, what is ticks? यहीं पे इसको जरा जवाब दे दें, टिक्स क्या होती हैं? आप इसको गूगल इमेजेस पे टिक्स क्लिक करेंगे ना तो आपको बहुत सारे इंसेक्ट्स नजर आ जाएंगे दैट कैन बी कॉल्ड एज टिक्स. तो मैं इसको आपको समझ में आ जाएगा कि मैं टिक्स कैसे कहते हैं, ठीक है? सॉर्ट ऑफ इंसेक्ट्स थोड़े से बड़े होते हैं साइज में, और आप देख सकते हैं. अच्छा, एक और बड़ा यूनिक ग्रुप है दैट इज कोलोबैक्टर एंड हाइपोमाइक्रोबियम. और यूनिक ग्रुप के सेंस में है कि आपने कभी अभी तक बैक्टीरिया में बडिंग नहीं सुना होगा. बडिंग इन बैक्टीरिया. तो मतलब आपने ये पढ़ा होगा कि ईस्ट के अंदर बडिंग होती है एज अ मोड ऑफ रिप्रोडक्शन, बैक्टीरियल केस में बडिंग होती है नहीं, ये हमने नहीं सुना अब तक. बट दिस ग्रुप इट हैज दिस यूनिक फीचर दैट they are present in aquatic environments. Definitely comparative to land, aquatic environments, they are less abundant with nutrients. So low nutrient aquatic environments such as lakes, and their feature is both of them, ये दोनों जो groups हैं species हैं, and they have stalks. They have stalks that anchor the organism to surfaces, means, and they attach themselves into two different surfaces by means of a stalk. So when the nutrient concentration is exceptionally low, so their mode of adaptation is that the size of a stalk is increases. So what advantage with this increased size of a stalk they have? Because when the size of a stalk increases, the surface area is increased, so the chances of nutrient absorption is increased. Right, I told you that they are the budding bacteria, because you know this fact that bacteria they divide by binary fission mostly. So but this organism is not dividing by binary fission, so they produce prominent prostheca. तो ये जो स्टॉक है आप देख रहे हैं, they are flagellated cells, but once they are attached to some surfaces, their flagella is lost, and their stalk it begins to form, it elongates. When it elongates, what happens? A small bud it starts to form and division begins. You see the meeting will end in 10 minutes. Okay, so division begins, flagellum forms on a new cell. ये छोटी सी बर्ड यहाँ से निकलती है, वो एक सेल के अंदर जो है वो आहिस्ता आहिस्ता वो कन्वर्ट हो जाती है, वहां पर एक न्यू फ्लैजिलम डेवलप हो जाता है और ये सेल पिछले से सेपरेट हो जाता है. So new cell with flagellum, it swims off as the swimmer cells, and stalk cells it initiates new round of replication. So this is a very unique form of reproduction in the bacterial case. Sabahat Umar is raising hand for what? Tell me. Okay. तो ये तस्वीर थी राइजोबियम, रेडी राइजोबियम, इसके बारे में आपने पढ़ा है, थोड़ा बहुत पढ़ा होगा. राइजोबियम इट नेम इंडिकेट it lives within the root nodules of the plants. कौन सी plants? Leguminous plants. So Bradyrhizobium is also the group that belongs to the same character. So थोड़ा बहुत difference है, ऐसी बात दोनों में है, so that's why they are forming different species. So Rhizobium, as the word rhizo, it indicates the roots of plants. So they are very much important agriculture bacteria, and they infect the roots of plants. Like infection means when some bacterial or viral organism, when viral particle or bacterial cell, it enters into any cell or it enters into any other organism, the term is known as infection. But not necessarily that clinical infection जो हमारे जहन में infection का concept है. तो यहाँ पर जो specifically it infects the root of leguminous plants means they are not causing different infections in the plant, rather they are residing in the plants. So to reside in any root of the plant, it first needs to be infected with that particular organism, right? So this is that infection. Entry of microorganism into any cell is known as infection. So common name is Rhizobia, and once they infect any root, they enter any root. So the characteristic feature is that the root forms nodules. So there is formation of nodules, and why they are making nodules? Because basically they are anaerobic organisms, right? So they live in the roots and they form nodules, and these nodules, they provide them formation of that nodules, provide them this opportunity to maintain an anaerobic environment where they can fix nitrogen. Because you know that these Rhizobium species, they fix nitrogen by means of an enzyme which is known as nitrogenase, and this enzyme is very much sensitive to oxygen. So that's why they need to have a place where oxygen is not available. So that's why they make nodules. So any nitrogen fixing organism, if it is not anaerobic in nature, or if it is not its enzyme is not sensitive to oxygen, it doesn't need in that case the nodule formation. So not every root infecting bacterial species forms nodules. So it is the requirement of Rhizobium that it forms nodules, because I told you the reason, that the enzyme that participates in nitrogen fixation, it is very much oxygen sensitive, so they form nodules. Agrobacterium, you heard this organism in your intermediate classes as well. This organism is of very much agricultural importance. And they also have this ability to invade plants, infect plants. But I told you that it is not able to form the root nodules or fix nitrogen, right? So Agrobacterium, basically it's a sort of plant pathogen. It can invade plants, but it is not fixing any nitrogen, it's not inducing any root nodules, and it forms a disease known as crown gall. The member of the species which is responsible for causing crown gall disease is the Agrobacterium tumefaciens, and the crown is that area where and the roots and stem they merge. So basically it's just above the surface. So wherever it infects, it forms a gall, a swollen part, right? So that's why it's called as crown gall disease. And this particular organism is very much attractive to different geneticists. Because this particular organism is equipped, or it contains a plasmid, and whenever it infects any plant, it enters its plasmid into the plant cell. So this information, it excites different biotechnologists or geneticists that yes, by using this plasmid, we can induce our gene of interest, or the gene of desirable characters into the plasmid of this Agrobacterium tumefaciens, and whenever it will infect that plant, that character will be expressed by the plant. So Agrobacterium has this much potential. Now, one thing more, that why this particular organism is very much important, because usually you know that it is easy to invade any animal cell because they do not have any wall, but plant cell wall is very thick. It is another lining surrounding the membrane. So if any pathogen is capable of invading that wall, means it has some potential. So that, and second advantage is that it also enters its plasmid into the plant cell, and the plasmid is also expressed in the plant cells. Its characters are expressed. So there can be this opportunity that you can remove those genes which are capable of causing infection in the plant from that plasmid, and you can replace those plant infecting genes with that of your desirable characters. So that's why the microbial geneticists, they're very much interested in these organisms. So now your assignment is that you need to search that what kind of characters till now has been transformed into plants using this particular organism. or its plasmid. ठीक है. Another important character आपने सुना होगा is Brucella, not character, group of organism. ये Brucella ये उन organisms में शामिल है जिसको हम कहते हैं कि it can be one of the dangerous, the most dangerous pathogenic bacterial species. So non-motile cocobacilli हैं, they are small and they are obligate parasites of mammals, or you can imagine that all the species, they are the obligate parasites, and they cause infection which is known as brucellosis. So it is one of the most, I told you, that pathogenic group. तो इसको बायो वेपन के तौर पे भी यूज किया जा सकता है. एंड एक और बड़ी इंपॉर्टेंट थिंग है, आपको पता है, अब जब आप इम्यूनोलॉजी पढ़ रहे हैं तो आपको पता है कि बॉडी का एक सिस्टम होता है कि whenever any pathogen enters your body, body starts phagocytizing those foreign particles. But Brucella is very much of medical interest that it can survive even the brucellosis, means it can replicate itself in the phagocytic cell. So basically your defense system is providing the opportunity for cultivation of this particular organism. So nature has given equal powers to your immune system as well as to microorganisms. So some of them they are very smart to use your immune system for the growth of their own self, for multiplying their own self. So brucellosis, they cause brucellosis in humans, and some of the symptoms of brucellosis are fever, chill, weakness, lethargy, muscle, joint aches and pains and headaches.